Hello everyone, I'm Paula Bray and I'm Director of Research for the Sydney Children's Hospitals Network. Welcome to our Advanced Therapeutics Webinar series where we focus on the rapidly evolving space that is advanced therapeutics and certainly the innovative and potentially transformational treatments this presents for children and families. And beyond. But before we get started today, I'd like to begin by acknowledging the traditional owners of the various lands that we're all meeting on today. The Sydney Children's Hospitals Network acknowledges that our facilities are built on the lands of the Bidjigal, the Caramagal, the Barramadigal, the Cabragal, the Wongal, the Wadi Wadi. Our services extend across the lands that are now known as New South Wales. Aboriginal people believe these lands hold the ancestral spirits of those who have come and gone before them. I acknowledge this was, and always will be Aboriginal land. I would like to extend my respects to elders, past and present, and extend that to any Aboriginal people who are joining us today. Today, we will be hearing from Scientia Professor Anthony Kelleher and Professor Leszek Lisowski. Professor Kelleher is the Director of the Kirby Institute at UNSW Sydney and a clinical immunologist at St Vincent's Hospital in Sydney. Professor Leszek Lisowski is the unit head of the Translational Vectorology unit at the Children's Medical Research Institute and a conjoint professor at the University of Sydney. Today, their presentations will focus on the translation of AAV and RNA gene therapies into the clinical setting. And following the presentations, as always, we'll move into a Q&A session to foster discussion. So please submit any questions that you have through the webinar via the Q&A tab, which is located in the bottom of your screen in the Zoom controls. If you see a question that you really would like answered, please also use the upvote by clicking the thumbs up beside it so that we'll make sure we can get to your question. But without further ado, I'd now like to hand over to Anthony to commence his presentation. Thank you and welcome.
Thanks very much, Paula. Just to make sure that this is going to share. That's showing up all right, I hope.
Looks great.
Thanks very much for this opportunity to present. And so today I'd like to talk about some RNA therapeutics that act as antivirals that we think provide platform technology. So as a background, there are two ways of classifying RNA therapeutics. One group are those that are dependent on the immune system, which we know well, such as mRNA vaccines that transform the prevention of serious COVID-19 infections, and for which there are now a number of products, including RSV vaccines. There are also self-amplifying RNA-based vaccines, and one of those that exists for COVID on the market. There are a bunch of other RNA approaches that are not dependent on mRNA and not dependent on the immune system. And they include short interfering RNAs, antisense oligos, and microRNAs. And today I'm going to speak mostly about short interfering RNAs. It's important to understand that these synthetic RNAs are quite different from the messenger RNAs that we've become so used to dealing with. They require very different production requirements. The attention of government is certainly on mRNA, but in fact, the greatest growth in therapeutic products is in the synthetic RNA space. And the FDA and the EMEA has approved more than 6 synthetic RNAs in the last several years, as against about 3 mRNA products. So how do these work? So they work on the basis of this RNAi, or RNA interference pathway, which is a highly conserved biological process mediated by small RNA molecules. And they mediate sequence-specific suppression of gene expression. And RNAi has 2 pathways, one of which most of us are very au fait with, which is post-transcriptional gene silencing, which works in the cytoplasm and it acts by these short interfering RNAs binding to target messenger RNAs in a sequence-specific fashion and degrading that mRNA target. It's very well understood. It has a short duration of about 3 to 4 days, but it's very prone to mutational escape as the matching of the base pairs in the target sequence has to be very exact. And we're exploiting this at the moment for coronaviruses, and we hope to exploit this for flu and RSV going forward. The other pathway, which is much less well understood, is called transcriptional gene silencing, or TGS. It occurs in the nucleus. The sequences that are targeted are in the promoter of genes. It works by inducing CpG methylation and changes in the histone structure architecture. So it's very much an epigenetic-based process. It's much longer lived and is in fact heritable through several generations. And we've exploited this to attack HIV, HTLV1, and human papillomavirus. So we've developed through a long period of time a way of developing these potential RNA therapeutics. The first thing we do is we look for highly conserved sequences across viral families. And we select the most highly conserved regions of those that have no predicted off-target effects, particularly no sequences that appear in the human genome, so that we're particularly targeting the virus, not the host. We then know that there are certain motifs within the nucleotide makeup of these targets that are more likely to work, and so we then screen those conserved areas for sequences that are likely to work for either the PTGS or the TGS pathways. We then screen those through reporter cell lines to shortlist the antiviral candidates, and then we, at least the last stage of the in vitro process, is to screen those against live virus in various models to identify the most potent siRNAs. And so through that pipeline, we've developed through to preclinical stage highly potent TGS acting HIV targeted molecules. We've developed, which will be the subject of the talk today, molecules that target SARS-CoV-2 and other coronaviruses, but less well developed down this pathway, similar RNAs against HTLV1 and HPV, which act by transcriptional gene silencing, and then a range of other siRNAs that target other viruses, including we've started to develop a range of other respiratory siRNAs that target a number of other respiratory viruses based on what we believe is approaching platform technology, which is based around these respiratory compatible lipid nanoparticles and this platform of low intensity nebulizer. So siRNAs are potentially powerful therapeutics, but there is a challenge in delivery. Firstly, these siRNAs are inherently unstable, though they are more stable than mRNA. There is no clear cellular uptake pathway that can be exploited apart from extracellular vesicles, and they're relatively difficult to make in a reproducible fashion with current technology. We've explored delivery of these through retroviruses and the retargeting of retroviruses to specific targeted cells, but this is pretty clunky and requires significant ex vivo manipulation in the laboratory. Other people have looked at the delivery of naked siRNAs without a nanoparticle protection. These are preferentially taken up by the reticular endothelial system and particularly by the liver, and are prone to cause inflammatory responses. You can package them with within LNPs, which certainly protect the siRNAs, but there is still significant preferential uptake by the liver. So a lot of work has gone into, at least when you're giving these things parenterally, to retargeting these to other sites outside the liver by varying the LNP composition, by conjugating them to monoclonals or to other ligands such as aptamers. But you can look at direct delivery of these to a site. And so where siRNAs have been very successful is the companies have exploited the fact that these are preferentially delivered to the liver, and they've targeted metabolic diseases in the liver that are genetically driven. And most of the products that have got through approval are in fact targeted to metabolic diseases within the liver. You can deliver these directly to the site by procedures such as ophthalmic injection, or you may be able to deliver these directly to mucosal sites, as long as you can find a non-inflammatory LNP and you've got a technique to do that. And that's something that we've explored and that I'm going to detail in the rest of this talk. So I'm going to tell you about a program which we call DARTS, which was the development of intranasal antiviral RNA therapeutics against beta coronaviruses. And this has been led by Chantal Allenstein, aided by two great PhD students, Ewan Zhang and Ellen Bowden-Reed, and with Scott Ledger, who's a postdoc in the lab. This was a large consortium of groups at the University of New South Wales, including the Kirby, the RNA Institute, and the Kids Cancer Institute. We worked very closely with Daniela Trainee at the Walcott and Macquarie University, and with colleagues at the University of Newcastle who are experts in respiratory epithelium and respiratory delivery. And then the in vivo mouse models came from Matt Johansson and Phil Hansborough. And we have an industrial partner from Holland called MedSpray, who make low intensity nebulizers. So what was the problem? Well, particularly at the beginning of the pandemic, there were very few antivirals against COVID-19, but we also thought that there was a necessity to have an approach to future coronavirus pandemics. The current antivirals are highly susceptible to viral resistance, rendering them ineffective. And this is best demonstrated by the failure of the monoclonal antibody approach. Many have side effects and limiting drug-drug interactions and have to be used very early in infection, such as Paxlovid. And then others like remdesivir need to be given parenterally. And as successful as the vaccine approach has been, there are always people that are going to be immunosuppressed or immunocompromised who will have less than ideal vaccine responses and will be more likely to get severe disease. And even today, the average hospital stay with COVID-19 is 5 days. So there is a significant burden on the healthcare system. And so we felt there was a need to build antivirals with a broad specificity that would target families of coronaviruses that were tolerable, that would hopefully lack drug-drug interactions and would have an ease of administration, and with a high bar to resistance to aid the current pandemic, but also potentially future pandemics. So our idea was to make a broad spectrum siRNA against SARS-CoV-2, but other coronaviruses as well, and that this would be delivered by inhalation protected by a nanoparticle. And so the mechanism that this diagram summarizes what we were approaching and what our approach was. And so what we've developed, what I think is potentially platform technology that I'm going to show you is we're developing for coronaviruses, but I think has potential to attack other significant respiratory viruses such as flu A and B, RSV, and unfortunately, with the growing lack of uptake of vaccination, perhaps even measles. So this platform technology, which we've called siRNA, it consists of three components: custom-designed siRNAs that target highly evolutionarily conserved viral sequences, an optimized LNP formulation that doesn't cause inflammation in the respiratory tree, and a low-intensity nebulizer that is tunable, that can deliver the nanoparticles to different parts of the respiratory tree, depending on where the focus of infection is, but doesn't distort the LNP product, which standard nebulizers do. So where we started with this is, as I said, we targeted highly conserved regions in viruses related to SARS-CoV-1 and SARS-CoV-2 across the animal kingdom. And we identified 514 potential conserved siRNA sequences. We put that through our algorithm, which I'm not going to describe, and identified 39 preferred candidates, which we then screened in vitro, and that narrowed it down to 16 siRNAs that were protective, and we identified from them 8 that we thought were lead candidates. And we further narrowed these down on the basis of potency and on highest sequence coverage across the entire databases of SARS-CoV-1 and SARS-CoV-2 to a smaller number of 6 lead candidates, which we prioritized for further testing. So we further narrowed these down to essentially 2 siRNAs, which we refer to as siRNAs 27 and 30, which target a region in SARS-CoV-2 called ORF1A, and they're located in this part of the viral genome here. And you can see that across, these are live virus assays, and across the strains of SARS-CoV-2 from beta through to Omicron, we get between 1 to 3 logs decrease in viral titer in a standard culture system with these siRNAs. When we look to encapsulate these siRNAs we started with some of the standard LNPs for delivery, and we started particularly with the Onpattro LNP based on the Onpattro formulation, which is used for the treatment of amyloidosis. And we got, these are the minimal criteria that we would accept for encapsulation, for size, and for something called polydispersity, which tells you about the shape of the integrity of the LNP. And we could demonstrate that we could encapsulate our siRNAs, both our control siRNA and our target siRNAs. We could get good, excellent encapsulation at a size and polydispersity that was acceptable using a standard nanoassembler. And importantly, for any of you that have worked with nanoparticles, reproducibility in this space is really, really difficult, we find. Not so much with LNPs, but certainly in other nanoparticles, and this process was highly reproducible. So we we chose these siRNAs for a number of reasons. They were highly potent. These siRNAs were compared to the other two siRNAs that we'd prioritized. These were about tenfold more potent in terms of their ability, the concentrations at which they knock down virus. And we could, when they were encapsulated in these LNPs, we could see effects down to 5 nanomolar in vitro cultures. Further, these worked whether they were applied to the culture pre-infection or post-infection. And you can see that this was 2 days pre-infection, so lots of times to degrade in the culture prior to infection, and they were still effective. And they were also effective if they were given after the infection was given time to establish itself. These siRNAs by themselves were designed in such a way that we found that, so one, they had no complementarity across the human genome. They were highly virus specific, and on screening assays such as the ability to induce interferon stimulated genes, they failed to stimulate these genes, and these in black are the positive controls. So there was no evidence of inflammatory responses with these particular siRNAs, and that was one of the reasons why these were prioritized. The LNPs were more difficult. When we used the Alnylam or Onpattro LNP formulation and exposed mice to these by intranasal administration, there was significant side effects in the mice. There was lung hemorrhage and a significant infiltrate of leukocytes shown here. We spent a lot of time reformulating this LNP, which I'm not going to talk about here, but this new LNP, this optimized LNP for respiratory delivery, failed to induce any inflammation, or so no lung hemorrhage and no inflammation, whether it was given as a single dose or in multiple doses to the mice. So we then proceeded to use that LNP in a challenge model with the people at the Centenary Institute using this mouse model, the K18 ACE2 SARS-CoV-2 mouse model, where the mice typically die over about a 7-day period. And we had improved clinical scores, lower viral titers, and lower viral PCRs in the lungs of these mice that were treated with the siRNAs, but not with the scramble control. We went on to look at whether these siRNAs in the LNPs could be delivered to where we wanted them to in the lung. And we had chosen, given the tropism of the Omicron variants and that tropism has been stable over the last 2 to 3 years, to target the nasopharynx. And so we tuned the delivery system, the low intensity nebulizer, to deliver aerosols in this range here, in the 10 micron to 10 nanometer range, that preferentially targets the nasopharynx. And we assessed it using this standardized industry accepted model of the respiratory tree. It certainly doesn't look like a respiratory tree to me, but this is what the industry recognizes and uses to assess respiratory delivery. And there are various sections of this that represent different parts of the respiratory tree, upper respiratory tree, and then lung here. So when we did this with Daniela Trinini's group out at Macquarie University, we found that this med spray device, as compared to two other devices, the AeroPump and the Philips InnoSpire, performed best in terms of looking at the yield of encapsulated siRNAs post nebulization. So we got good yield of encapsulated siRNAs as expected with this device, but not with the other two devices. You can see there's big differences here between those numbers, about 80% here and 50%, 50 to 70% here, and about 10 to 20% here, yield of encapsulated siRNAs. Furthermore, when we looked at where these were delivered, they were delivered to where we expected them to be. The purple is the nasopharynx, and so these were delivered, the majority of the nanoparticles were delivered to where we wanted them to be, as tuned. And this is the device here. So you can see that it's very small and is portable and can be could be used very easily in a range of situations, so very unlike a standard nebulizer. Furthermore, the delivery through this mechanism maintains the integrity of the siRNA, both in terms of its size. It maintains its expected size at less than 100 nanometers, and it has the expected polydispersity after it's been put in after it's been delivered to the nasopharynx. So the final experiment we did in this group of experiments was we took this model of bronchial epithelial cells called Calu3 in a multi-layer culture which we grew in snap wells. And then we put those cells within this model of an olfactory bulb in a different machine to the one I showed before, that again is an industry standard. And then we applied the siRNAs through the nebulizer using the med spray device into this machine, and then rescued the cells and took the cells back to the PC3 lab at the Kirby and exposed those cells to virus in the lab. And you can see that the siRNA treated cultures shown here in green were it suppressed virus. So the packaged siRNAs delivered through the nebulizer to the area we wished it to be delivered to was effective in suppressing virus. So what I think we're approaching here is platform technology for RNA delivery to the respiratory tree. We've got now we've got siRNAs targeting two highly conserved regions of coronaviruses that are highly resistant to mutations. And because they're pan-beta coronavirus, they should not only be effective against the current SARS-CoV-2 virus, but potentially to future coronaviruses. We have what we feel is an optimized LNP that's safe and non-inflammatory for delivery to the respiratory tree, including the lung. We've shown in an in vivo model that this improves clinical outcome and reduces virus in the lung. We can deliver the intact siRNA LNPs without shearing and with functionality to the desired region of the respiratory tree, in this case, the nasopharynx. But the device is tunable and we can deliver that to other areas. Because this is topical, We think that there will be fewer side effects and fewer drug-drug interactions, and this is an easy way to administer this that could be used either in a range of settings, whether that be inpatient or outpatient, and pretty much regardless of age as well. So this is now progressing to formal toxicity studies and down a drug development pathway for regulatory submission. Hopefully, if it doesn't fall over, we'll move to clinical trials. And in parallel with that, we're exploring a similar approach for flu A, flu B, RSV, and sadly, thanks to the US administration and the anti-vaxxers, measles. And so a large number of people who were acknowledged on the slides as we went through have contributed to this. I'd also like to acknowledge Stuart Turville, who provided the live virus in addition to those people, and just acknowledge that this was all led by Chantal Allenstahl. And I'll leave it there. Thanks very much.
Thanks, Anthony. That was fantastic. We'll hold questions till the end, and I'd like to welcome Leszek up to provide his presentation. Thank you.
Okay, I assume it's visible.
Yes.
Thank you very much for giving me the opportunity to talk about what we're doing in my lab in the context of AAV-based gene therapy. Oops. I'd like to start with this slide just because I may run out of time, and obviously this is the most important slide. It is my team at CMRI that actually performed the research I'm going to talk about, as well as our funding sources. So I'd like to start with a big picture and just kind of and then zoom in on the solution. So let me start with the problem. Not many people realize that the genetic disease impacts more people than HIV and cancer combined. There's an estimated of 10,000 genetic diseases that affect every year over 8 million newborns. And the saddest part of the statistics is that about 30% of those children will not live long enough to celebrate their 5th birthday. Now, there are some exciting times. Over the last couple of decades, we've witnessed 3 very important revolutions. Recombinant DNA revolution, which allows us to create preclinical models and disease models. RNA-DNA sequencing revolution, which enables us to sequence patients and diagnose them very rapidly and precisely. And more recently, genome engineering revolution, where we can now go into the genome and modify them inside of the patient cells. But the question is, what is actually the medical impact of all this progress in genomics? And I like this simplified slide I borrowed from Ian Alexander that illustrates that the concept that our knowledge is growing exponentially. And that's definitely obvious in the last few years with all the AI that came in. Our ability to diagnose diseases follows a very similar path. But when it comes to prevention or treatment, there's this gap that has formed. And this is why I believe the gene-based advanced therapies have a very important role to play. Now, this brings me to the concept of gene therapy. And gene therapy is simple. It's using gene as therapeutics. So you can imagine having a therapeutic gene. This is the medicine. We package it in a sort of a syringe, and I'm going to talk about viral vectors, but I'm also going to mention non-viral vectors, and we deliver that to the patients. It sounds a little bit sci-fi, a little bit something from a movie or so, but it's not true. It's actually already happening. There's 7 drugs today on the market using the exact viral vector that I'm going to talk about, called AAV. And this is just the beginning. The 7 products I showed you are in the middle of the circle here. Those are the marketed drugs. And you can see this as you keep going towards the outside of the circles. Those are the phase 3, phase 2, and 1. You can see that there's this avalanche of programs using those technologies, and they are really changing the healthcare system. But despite the recent advances in this field, there are still problems with gene therapy. And I really like this famous quote by Inder Verma, who said that there are only 3 problems with gene therapy, and that's delivery, delivery, and delivery. It isn't going to be a problem to make gene therapy work if we have an appropriate set of tools, and that's the key part of the sentence. But without them, we don't have a hope or a prayer. Now, the delivery methods can be broadly divided into physical-chemical, such as LNPs that we just heard about, or PEI. Those are different lipids and chemical-based methods to deliver the DNA or RNA to the cells, and viral vectors. And there's many different viral vectors, but I'm going to concentrate on the AAV. But I believe that it's really the viral vectors that became the gateway for gene therapies, so allowing it to be where we are today. In my lab, we utilized a virus called adeno-associated virus. This is this little guy here on the picture on the left. It was discovered as a contamination of an adenoviral culture, hence the name adeno-associated virus. It's a small and quite simple virus, two main open reading frames, rep and cap, encode 7 main proteins. The proteins that are going to be of main interest today is the VP1, 2, and 3 that form the structure of the capsid. So when they come together, they form this beautiful structure shown here on the bottom, where each of the sticking parts of the protein would actually contribute to how the virus behaves or the vector behaves inside of the cells. Now, for AAV, another concept that's very important is pseudotyping, or sometimes called pseudoserotyping. What it means is that we can take the AAV genome, and just by switching one plasmid during the manufacturing process, we can put that genome into different capsids. So we can very easily cross-dress the AAV genome into different protein shells. And that's important because different capsids have different properties. The main one is tropism. So they serve like a pill, coating of a pill that's going to deliver the cargo to different therapeutic targets. But in addition to the tropism, the capsid influences how easy is the vector to manufacture and how the immune system is going to see it. And in my lab, we take 3 approaches to developing new vectors. The one I'm going to talk about today is bioengineering. And bioengineering, there's many methods. One of the methods we use is called directed evolution. And directed evolution, as the name implies, it mimics the evolution, so the process that happens in nature, but we can direct it in a very specific way. So we all know that the evolution works through random mutations that happen in the organism, then combined with a selective pressure, let's say a deeper flower, select for a new feature. And this is sort of what we try to do in the lab. So we create what we call AAV libraries. Those will be the populations of AAVs that have all the possible mutations that could happen in nature. We then apply selective pressure, let's say transduction of a organoid or transduction of a lung. And through that, we can then select a new AAV with the properties we are after. And one of the methods there's many methods we use to create those libraries, but the method I want to talk about today is the one based on AI or machine learning. And it's a very powerful method. And if you asked me about this a few years back, I totally didn't believe it, but I was proven wrong by my postdocs. So again, this is AAV capsid. And if you look at those loops that you can see, those purple loops on the outside, this is what would then interact with the receptor on the cell surface. So one of the ways we can engineer AAVs is by putting new protein loops, so short peptides shown here in red, on a surface of the AAV. And I'm going to use example of 2 such loops, one of 7 amino acids and one of 10 amino acids length. Now, if we want to make a saturated library, so every single possible combination of 7 amino acids, that actually is 1.28 10 to the 9th variants. And that's represented by this large reddish orange cube. So this is our theoretical space of all the possibilities that nature gives us. But what we can comfortably propagate in the lab and actually test is represented by this, and then this is to scale, by this little yellow cube here on the bottom, and that's about 10 to the 7th. So we are probing about 1% of all the possibilities in our experiments. And that's for 7 amino acids. If I go to 10 amino acids, and again, the large cube is to scale. So now we have 10 to the 13 variants. That's the possible, all the possible combinations. A little dot here in the bottom is the actual space. We are now about 6 logs, 1 million times off from where we're supposed to be. So what my postdocs, Marty and Suzanne, wanted to prove is that we can use AI to help us, kind of probe the whole theoretical space. And they started with a simple experiment. They created a library, and they asked computer, a custom-made algorithm, to perform selection of the library and select for AAVs, new variants that can transduce those cells called H287. Very simple in vitro experiment. And at the same time, they took the same library and performed physical selection in the lab. So the physical selection took about 6 weeks, and obviously people had to be involved and do the work, versus the AI-based selection took about 2 weeks. Now, at the end of the process, we chose 10, or they chose 10 top AAVs from both selections, and they tested their performance. So this is the 10 AAVs tested individually from the physical selection. You can see that there's some good ones that can transduce over 40% of cells at this tested dose, but some didn't perform so well. This is what the computer selected. Now, they're not better, but you can see that virtually all of them worked, and no labor was involved. It really was 2 weeks of the computer crunching numbers. But this is very simple. This is in vitro. So then they went on and did the same thing in vivo. Here they did not only in vivo, they also increased the bar a little bit higher. The selection was done for packaging, so make sure that the vector can be produced. And they want to select vectors that transduce liver but don't transduce kidney, or transduce kidney but don't transduce liver. Long story short, they selected 5 top AAVs. They all produce well. The ones solid lines is the one that was selected for kidney. So you can see they don't perform so well in liver, but they perform better in kidney. And vice versa, the dotted lines are the AAV selected for liver, but not to work in kidney. And that's what happened. So the idea here is not to use computer to actually select the final AAV that's going to move to clinic, but more to allow us to take this theoretical space of possibilities and choose the best candidates to make a smaller cube in a way that then we can probe experimentally and polish the experiment in the lab. Now, the caveat here that some of you may know is to do this work, you actually have to train your algorithm. So in a way, you have to perform the experiment first, generate the data, teach the algorithm in order to be able to generate the data. That sounds a little bit like a chicken and egg, and it is. But what we actually are getting better at doing is performing the testing, the training experiment in a simplified model, and then extrapolating from that so that the final selection can be done, let's say, for a human. So you can use a mouse to train and then a human to select. Another very important aspect of when you do directed evolution is the model in which you're going to select. And obviously, this is a very simple picture. On the left side, you have tissue culture cells, easy to use, but very far removed from human, which is the ultimate target. The closer you get to human, the more difficult it gets, but it gets you closer to the final target. In our experiments, when it comes to liver, we use very often humanized mice. Those are mice that can be repopulated with primary hepatocytes from human or from monkeys, so we can humanize the mice or monkeyize them. But more recently, in collaboration with researchers from Royal Prince Alfred Hospital in Sydney, we started using liver explants. So now we can take a human liver, the entire organ, and keep it alive for a number of weeks, and gives us a great opportunity to develop and validate our AAVs. So this is the first study that we published just last year, where we took a number of AAVs shown here on the top, and we screened them in the liver explant. And we selected a serum that was plasma, that was used in this experiment not to have any antibodies against the AAV. And you can see that there's different performance of different AAVs. This is based on next-gen sequencing, so it's relative performance. But we were able to identify 2 of the capsids that actually we developed, Sydney 11, Sydney 12, as a top performance. In the follow-up experiment, we actually chose serum that had antibodies. And you can see the neutralization profile on top. And long story short, is the middle of the panel gets removed. Those are the AAVs that got neutralized. So not surprisingly, they didn't perform well in the experiment. But there's a few other very interesting things here. First is if you look at AAV5, AAV5 does quite well at the DNA level. Again, this is relative, but it does best from the whole panel. But despite that, it doesn't express well. So on the lower panel, you have a RNA or cDNA and the DNA cell entry on top. And why is that interesting? Because AAV5 is actually an AAV behind 2 marketed products. And it's not a very good AAV, but it is resistant to antibodies. What we also showed us that we were able to identify some of our bioengineered vectors as more resistant to neutralization. So again, not a big surprise, but we realized we have to pay a little bit more attention to the neutralization antibodies when developing AAVs. So there was a few things that were happening in the lab. In Ian's lab, there was an ongoing project where they purify B cells from patients treated with gene therapies, then amplify the monoclonal human antibodies that were developed in patients against AAVs, and they map them on AAV surface in collaboration with a team from Florida. Our group was more looking bioinformatically on which part of the capsid could be involved in some of those interactions as well. And Marty identified one of the regions, which is called VR1, and created a saturation library of this region, which gave you a library of almost 15,000 variants. It's actually quite a small library. And then he performed selection of AAVs, and this is the first time we did selection in primary human explant. And I'm not going to spend too much on this slide here, but I just want to show you that this data is normalized to Sydney 12, which was the best AAV on the previous slides. And those are the AAVs that we selected in this experiment. And you can see right away that we are, significantly better, up to 10 times better at the DNA level, at the RNA level, in the presence of neutralizing antibodies. Why is that even more interesting? That if I normalize this data to LK03, LK03 is the first bioengineered vector that actually Ian and I co-developed when I was a postdoc, and it's in clinic now. We are actually up to 700 times better than LK03. And LK03 is doing very well in clinic. And why is that important? Because if I go back to the slide that I showed you, the 7 drugs that are on the market today using those technologies, those are the price tags at which they enter the market. This is obviously single dose, but this is US millions. Now, if we could shave 2 zeros out of this by using better vectors that then allow us to use lower doses, that would be significant. That would be a game changer in this whole field. Everything I showed you right now was based on liver. We are a technology lab, disease agnostic to at some level, and we're using performing similar studies in many different organs and tissues. Again, trying to develop technology that then ourselves or other people can take and bring to clinic. But why are we doing this? I mean, obviously the technology can be a game changer and it's going to have an impact. But why do we do it? It's a tough job, highly disappointing. What drives us to wake up in the morning and do it? And this is some of the smiley faces that actually make us do it. Those are some of the patients we work with that we try to develop gene therapies for. And those children are affected by rare diseases, which is what my lab specializes in, developing therapies for rare and ultra-rare diseases. And that brings me to the concept of Australian Genome Therapeutics Center, which is a name that I've been using and something that I've been working with key decision makers on campus to establish. And it's a virtual thing. It's just in a way bringing together the expertise and infrastructure to allow us to take a patient from detection to correction. And I know it sounds a little cliche, but it's actually very important, especially on campus when we have amazing research facilities and a specialized hospital that can bring those therapies to the market. To the patient, sorry. What was missing, however, was the manufacturing facility. And recently, with help from the New South Wales government, we were able to establish Australian first clinical grade viral vector manufacturing facility. And that is very important because when we look at the costs of developing therapies from detection to phase 1-2, about half of that cost is actually the manufacturing itself. So with that, I'll briefly introduce viral vector manufacturing facility that is being established on campus. It's a 3-stage process. The first stage exists already that allows us to produce vectors up to 25-liter scale. The second phase is almost finished, almost completed, and that will bring us up to 500-liter scale. And then the third phase will be a greenfield that will allow us to expand even further as the needs increase. Now, my lab is taking full advantage of this amazing expertise and infrastructure that we have here. And this is just a list of some of the diseases we work on. And again, you can see that the names are weird or funny because they don't have names. They're so rare that they are named by the name of the gene that's mutated. CTNNB1, which I'm going to use as an exemplar, has about 500 patients that we know of. PGAP1, 25 patients. So in the whole world. So you can see those are, not diseases that a regular pharma is going to pick up. So let me just introduce the story on using CTNNB as an exemplar. So it's a severe and rare neurodevelopmental disorder, first discovered or described in 2012, so not such a long time ago. And it affects about 1 in 50,000 individuals. That's how we estimated the rate of the frequency, sorry. The program actually brought together researchers from all over the world. It was initiated by a mother from Slovenia. Not surprisingly then that there was a lot of researchers in Slovenia and neighboring countries, but there's also Hong Kong and obviously Sydney. And we are not trying to elevate the bar too high. We're trying to do it as quickly and as efficiently as we can. And in this context, we take advantage of Zolgensma. Zolgensma uses AAV9, which is an AAV that can be given systemically, crosses blood-brain barrier, and delivers the cargo to the CNS, to the brain. Or it can be also given directly into the brain. So pretty much on the bottom here, you have a cartoon representation of the therapeutic cargo in Zolgensma, and we're trying to minimize the change as little as possible to convert Zolgensma into a new drug. This is just the highlights of the project. We started because we were, for the first time, expressing this transgene from a vector. We tried many different combinations. We then developed preclinical models. There was a mouse that we could actually use. Eventually, we narrowed down from the 7 constructs that we started with. We chose the lead candidate, went into manufacturing in Spain. Our facility wasn't ready yet. And now in 2025, we are finally ready to initiate the first clinical trial, which is going to happen in October in Europe, followed by the secondary site here in Australia. But this briefly, also at the end of the talk, I want to briefly talk about, in general, the rare and N of 1 disorders. So I mentioned that there's approximately 10,000 of those disorders that combined affect 300 to 400 million people globally. On average, 1 in 10 children is affected by one of those diseases, and most rare diseases affect fewer people than 1 in 2,000. Actually, 85% affect less than 1 in 50,000, and some conditions affect a single child, a single person worldwide. Because of that, the traditional drug development is very expensive and requires large patient population, and it's not fit for rare diseases. So new translational solutions optimized for rare disorders are required. And this is a 2022, I believe, paper so it's kind of small, but it divides diseases into common in green, rare, ultra rare in blue, and then hyper rare. And you can see that, and then on the x-axis, you have a number of diseases that would fall into this group. So you see that the rare diseases, the rarer it gets, the more types of diseases we actually have, and combined the number of patients affected is also actually larger. So what I believe is, and I think this is also common knowledge right now, is that while big pharma is quite interested in the common indications, where obviously there's a big market and they can make quite a lot of money, the larger group of populations is shown here in the green box, are left for academia, philanthropic, and non-profit organizations to tackle. And this is a slide I borrowed from one of my PhD students, whose picture I forgot to put here. He came up with this gene therapy ecosystem. So when you look at all the drugs that have made, all the gene therapy drugs that actually made it to market today, they all started in academia, which is where obviously the knowledge is initiated. Then they went into biotech companies, and then they increased the scale and the big pharma. So this is kind of the path that those programs take. And I believe that when it comes to rare diseases, this is changing, and actually it's a little bit more flat. It's really going to be between academia and then philanthropic, non-philanthropic, non-profit organizations that are going to fill up the role of the biotech and big pharma. But even with that, there's still questions that remain outstanding. How and where do we set up the phase 1-2 trials? How do we enroll enough patients? How do we ensure that there's enough patients to even conduct a trial? Do we actually aim to bring those therapies to the market? I mean, do we want to? How do we do it? Is that even the path that we want to take? Who is going to be the sponsor of those trials? Who pays for it? Can we find a commercial partner? Do we actually want a commercial partner? A commercial entity can pick up a program and then decide that it's not profitable and drop it. And those are very expensive programs. How do we ensure that what we develop is accessible to everyone, regardless of their geographic location and socioeconomic status? And also, can those programs become self-sustainable? Can we come up with a model where actually they create enough income or profits to feed the next program, and so that this can continue and benefit more children? So there's many different solutions that people are looking at. This is a solution that I came up with my colleagues. We set up a foundation, Gene to Cure Foundation. It's a US-based foundation that then established an Australian-based for-profit company. This allows us then to raise money through the US foundation, perform the research, the R&D activities, which is the research, manufacturing, and clinical translation, the phase 1-2 in Australia, taking advantage of the R&D tax. And then once the project is de-risked and validated, it comes back to the foundation that then raises funds to bring it to the patient. So again, it doesn't solve all the problems, but it is, I believe, a step forward. And I want to finish with the first slide I started, the problem. There's a lot of diseases. They affect over 8 million children, and many of them die within the first 5 years of life. And it is tragic, but I hope I showed you that the technology has reached this level of maturity, and we're also doing our part to change the statistics for the affected patients. Thank you very much.
Thank you so much, Leszek. And if I can just invite Professor Tony Kelleher to please put his video camera back on just for the last few moments. My name's Dr. Michelle Lorenzos, and I'm the Advanced Therapeutics Medical Lead at the Sydney Children's Hospitals Network. We're incredibly grateful to both of the experts today for giving their time. There's probably only a few minutes left for questions. I know it's difficult to summarise huge bodies of work in the 20-minute timeframes you've been given, so it's not unusual for time to go over. But I guess I might just take the liberty of asking a question, given that we don't have too much in the Q&A box at the moment. And really, I'd like to just be forward thinking. So often when I attend scientific conferences and we talk about gene therapies or RNA therapies, there is a recognition about affordability and access. And Tony, in particular, when you speak about infectious diseases, it makes us think about populations for whom, very expensive drugs and treatments may just not be tenable. So what could you say about, I guess, the future of these technologies in terms of affordability and access, and where do you have some hope or optimism about ways of getting treatments to people who need them in a way that's accessible?
So it's a big challenge, and it's a very good point. So the way we're going to try and develop these is that we've got a not-for-profit company, and they will. That we're working with. They have already taken drugs to market in low and middle income countries. And so they have the expertise in there, and they have the same desire to do that as we do. So what we would do is they would have first refusal for the development of these, for the licensing of these in low and middle income countries, which we would do at a rock bottom price, and then try and license them in the first world for more money, so that the delivery of these to low and middle income countries would be at cost or better.
Yeah, I will take a stab at that question as well. It's important to realize that those technologies are expensive and difficult to develop. So that's always going to be expensive, and they are also expensive to produce after all. So afterwards, it's not a small molecule that you can just make for a cent at a pill. However, I'm hopeful because some of the pricing is driven by biotech trying to recover the lost cost. But as the technology gets better, so in AAV-based therapies, the vectors get better, which means the doses that are required will be lower. But also the manufacturing process, a lot of the processes involved and that drive the price are also getting better. Simply, gene therapies are still new entrants to the market. We've talked about them for the last 50 years, but they really didn't work. So now that they do, there is a pressure to improve the manufacturing technologies, which previously we didn't really invest time into. So I think the gap is going to get bridged by lower doses, better technologies, but also decreasing the cost of manufacturing and other important aspects of the whole development. So, but I don't think they're ever going to be super cheap, but I think they're going to be more affordable.
Thank you so much. And it's positive to hear about how we can first of all work on making sure we get the right treatments for these diseases, and then think about how we get those treatments to the right people at the right time in a way that's affordable and accessible. And on that note, I'd hand back to Paula Bray. Thank you.
Thank you. And unfortunately, we've run out of time, but I really just want to say an enormous thank you to both Anthony and Leszek for those excellent presentations today. Just a reminder for everyone online that a video of the webinar will be available via our website and the Sydney Children's Hospitals Network YouTube channel. There'll also be a short survey at the end of the webinar, which will automatically open in your browser when the webinar ends. So if you have a moment to fill it out, we'd really appreciate your feedback. Again, thank you so much for another incredible webinar today. I would just like to acknowledge our contributors, importantly the Luminesce Alliance, and again, thank you all for being here today and for your time. And please stay updated for our next webinar by subscribing to the Research Interest Group mailing list or the QR code on screen. Thanks so much, everyone, and bye for now.
Thank you. Bye.
Thank you.